don't mind, I will turn my video off because also my internet connection is lagging. So I don't want it to buffer during my presentation. Okay, thank you. Okay. Just a second so I can share my screen. Can you see my slides now?
Yes.
Yes.
Okay, great. So, hi everyone. My name is Nisma Adbraman. I'm a genetic research intern at the IWI Foundation, and I'm currently working as a full-time genetic laboratory technician at a medical laboratory where I do a variety of genetic tests. I live in Riyadh, the capital of Saudi Arabia, and I'm very excited to do my presentation on gene therapy for trisomy disorder today. I hope you will enjoy it. So trisomy disorders are chromosomal abnormalities characterized as changes in the number of autosomal chromosomes, typically an extra autosomal chromosome. This extra chromosome occurs due to failure in chromosomal separation during meiosis. The most common trisomy disorder is trisomy 21, known as Down syndrome, which is the most common type of trisomy disorder, followed by trisomy 18, known as Edwards syndrome. And on rare occasions, a pregnancy with trisomy 13 can result in a live birth and give rise to a baby with Patau syndrome. The severity of the trisomy condition is predicated on the extent to which the extra chromosome was copied. If it's a full trisomy, it means the entire chromosome 21, 18, or 13 is copied. And individuals who suffer from full trisomy have more severe symptoms. If only one part of the chromosome is copied, then it's called partial trisomy. And the symptoms of partial trisomy are usually less severe than full trisomy. With the knowledge that the extra chromosome expresses its genes and causes trisomy symptoms, to mitigate the effects of this extra chromosome, scientists looked for ways to silence this extra chromosome by studying the gene regulatory processes that control genes expression and occurs naturally, and how to use these processes to regulate the gene expression of the extra chromosome. One of these processes is called X chromosome inactivation. In female mammals, chromosomal silencing occurs naturally and randomly. X chromosome inactivation is when one of the female X chromosomes is inactivated. The inactivated X chromosome is tightly packaged into a structure called heterochromatin. When a chromosome is in a heterochromatin state, it cannot be transcribed and its genes won't be expressed. This is a random process. One of the chromosomes in females is inactivated, and there is no process to determine which X chromosome will be inactive in any given cell. One of the visible manifestations of X chromosome inactivation is the fur color pattern in female cats. For example, the orange patches of the cat fur you see in the picture are the result of the inactivation of the X chromosome, which carries the allele coding for the black color in those particular cells. And in the black fur patches of the X chromosome, which carries the alleles coding for the orange color, is inactivated. X inactivation occurs naturally early in development, and the chromosome remains inactive for the rest of the female's life. One of the genes that control X chromosome inactivation is called XIST gene. The XIST gene located on the human X chromosome and exclusively inactivates one copy of the X chromosome in mammalian females with XS karyotype as a natural mechanism to adjust the gene dosage between females and males. Because females carry two X chromosomes and have double of X chromosome genes compared to males, this inactivation even gene expression between males and females. Inactivated X chromosome manifests as bar bodies that can be found on the periphery of the nucleus in cells. So what are the gene therapy methods we can utilize in order to silence the extra chromosome in trisomy disorders? There are two potential gene therapy options for treating trisomy disorders. One is using gene engineering technology called zinc finger nucleases to insert XIST gene early in development to use its chromosome inactivation ability to silence the extra autosomal chromosome. Zinc finger nucleases are enzymes that target a specific sequence of the genome and cleave the DNA at that specific sequence, and then use the DNA repair system to insert the targeted gene into the patient genome. The targeted gene in this case is XIST. In one study where they used Down syndrome stem cells, the zinc finger nucleases enabled the accurate and efficient insertion of the XIST gene into chromosome 21 in Down syndrome stem cells. And gene expression in the Down syndrome stem cells was identical to that in the normal individual stem cells. Another potential gene therapy for trisomy is using CRISPR-Cas9 to introduce breaks in the targeted chromosome in order to silence it. Multiple RNA proteins guide the CRISPR-Cas9 system to the targeted sequence of the chromosomes. These RNAs identify repetitive sequences in order. In one study, they used the repetitive sequence of the centromere and long arm of the Y chromosomes in mice. This resulted in the elimination of the Y chromosome and resulted in mice with XO karyotype. They also found that using RNAs targeted repetitive sequences of chromosome 21 in Down syndrome stem cells converted trisomy 21 to disomy at a success rate of approximately 15%. Even though these options sound very promising, there are some challenges we need to address to implement gene therapy optimally. Some of the challenges are that, first, gene therapy for trisomy is still in the experimental phase. These studies were performed on stem cells and animals, and it will be a long way until we reach the human trial phase. Another challenge is the off-target effects of gene editing technology. Gene editing methods such as zinc finger nucleases and CRISPR target specific sequences of the genome. As scientists design guide RNAs that can identify the target sequence as precise as possible, but the potential of gene editing tools targeting the wrong sequence and causing adverse outcomes remains a big challenge, hindering gene therapy in general. And lastly, one of the most challenging aspects of gene therapy is the narrow developmental time when gene reprogramming should occur, and the ethical implications of doing genetic engineering at such an early stage of development. Is it ethical to alter the genome of embryos or babies? And if the door is opened for altering human embryos, what are the lines we should draw, and who gets to say, okay, these are the characteristics we should eliminate, and these are the characteristics we should add to the human genome? And lastly, this is a reminder to be mindful and listen to the concerned voices of advocates who worry about gene therapy as a gateway to imposing an ableist status quo, wishing to eliminate any differences deemed unfit for society, disregarding disabled individuals' autonomy and wishes, and to also respect the broad spectrum of human lived experiences, and keep in mind some people see their disability status as part of their identity. And to conclude my presentation, I hope that what you get from it is that gene therapy is an exciting option for treating trisomy disorders. Although the field of gene therapy is still in its infancy, we should not overlook its potential for reducing suffering and improving lives. And here are my references. thank you very, very much for listening to me. And if you have any questions, I will be glad to answer them.